Today, Charles and George will tell us particularly about the ontology that has been scientifically expanded to now incorporate the structures in the brain of 3 species, soon to be more than that, or 4 species actually, because we need to count the chicken in as well, I think.
Yep.
And with no further ado, I will hand over to Charles. You're starting this for the seminar.
Thanks, Johannes. The key word here actually is the Puelles mammalian brain ontology. It's not that Louis Puelles has done it all, but he's certainly been a central figure in the work that has basically demonstrated the gene expression showing the embryological origin of different parts of the brain and which ones have features in common and where things split, which actually gives you a tree in the end, an ontogenetic tree which underlies the ontology. And for those of you who don't know, Louis Puelles is in the University of Murcia on southeastern Spain, and he's basically the best neuroanatomist in the world, from my opinion. He's a traditional medical school neuroanatomist who's a master of gene expression and comparative vertebrate anatomy, and we've benefited enormously. In fact, this whole project has come out of our association with him. It started about 11 years ago when Nicole Edouarin in France, whom I'd worked in a lab for a while, basically collared me and said, look, you've done these terrific rat brain atlases and other things, but we work in chicks and we need a chick brain atlas, and when are you going to get on with this? And George and I really knew nothing about chick brains at that particular point, so we said, well, we'd have to have someone who actually knows about chick brains, and we can make atlases, but we know. And eventually they introduced us to Louis, and Louis came out to Sydney to start the work with this, with his wife who'd prepared these beautiful sections. And I remember distinctly the first day that we sat down, George and I, fairly painstakingly work out boundaries and try to put lines around, and we argue a lot. And Louis just picked up the pencil and he said, okay, this is the hypoglossal nucleus, and this is the nucleus of the solitary tract, and this is, and he just ran one nucleus after the other in the chick. Well, we just watched, and we didn't say anything for really about the first hour as he ran through this, because the other thing is he also gives you a commentary. He says the dorsal motor nucleus of the vagus in the chick is actually mostly called the glossopharyngeal nucleus, and it's known to have a number of subdivisions. And he gives you a little commentary on the history and connections and so on. So we realised that we were actually in the presence of something rather different than we dealt with before. And the thing that amazed us after working for a while was that Louis knew what should be arranged with what, because he'd been doing all of this in situ hybridization and gene expression for a long time, which we hadn't done. We were complete novices. I mean, we'd done a bit of histochemistry and that kind of thing, but not gene expression stuff. And so it slowly dawned on us that actually there was something much more we had to do with the Atlas system to enable things to work out logically. And of course, just at that time, people were starting to press hard for an ontology which might be an alternative to the traditional ontologies. And Doug Bowden has done a fantastic job in creating a complete ontology name system for mammalian brains over a long period of time, and it's solved many problems within that system. But what this showed was that gene expression patterns would give you a different view of that, and one which one can argue on the basis of, because it's ontogenetic, because it's developmental, might give you a truer picture of the precise relationships of the different parts of the brain. And so during the production of the Chicken Brain Atlas, George and I learned more and more about the principles which were involved in interpreting gene expression data, and I guess it was really a bit of a surprise to me when the book was actually complete. Louis just added in at the end of the introduction the complete chick brain ontology, and that is the only comprehensively published version of the new vertebrate brain ontology, I suppose. It's in the Chicken Brain Atlas. Chicken Brain Atlas is not and will never be a great seller, but it is probably the most, how can I put it, the most sophisticated vertebrate brain atlas that you will encounter. While our rat brain atlases have some great features, the chicken brain atlas has the complete story of the ontology embedded in all of the pictures that are in it. If any of you have not seen it, you should have a look. It's even if you don't work on chicks, it's such a fantastic source. Not least because Lewis wrote an 80,000 word introduction. I was so embarrassing. I drafted out some headings for an introduction because I was trying to push the project ahead. And I sent it to Lewis and said, just fill in these bits and, we'll kind of get it all together. And he said, Charles, he said, you completely misunderstand what we're doing with this. I am writing an introduction and it is not going to be 10 pages long. And so anyway, the introduction does provide a wonderful background to the understanding of vertebrate brain organisation and looking carefully at the history, why we've been led in certain directions and how we've managed to ignore quite important things. I mean, one of Lewis's other talents is that he grew up in the Canary Islands, part of Spain, but where there was a German colony, so he grew up speaking German, so he reads all of the old German manuscripts, Zien and Hiss and those sorts of things, and in fact Hiss, the work of Hiss really presages all that's been found in the modern ontologies. Hiss had it all worked out. He knew about rhombomeres. He had the isthmus identified. He had the thalamus and hypothalamus appropriately located, and so on. And so that was a revelation. And of course, just as finding people like Hiss had indeed gotten a different sort of elaboration, it's also important to note that the field was dominated from about the 1910 onwards by the work of the American Herrick. And Herrick quite properly emphasised the alar basal organisation of columns within the hindbrain, which he extended up into the forebrain, which is where the trouble starts. Down in the hindbrain, the alar and basal separation works very well. You have the motor things from the basal plate and the sensory things from the alar plate. The problem is that if you consider the brain to be essentially a longitudinal cylindrical structure and you take it through into the forebrain, things start to go wrong. And the first wrong thing is that if you do this, you conclude that the thalamus is kind of an alar structure and the hypothalamus is just the underneath part as a basal structure, and that doesn't work. The reason it doesn't work is that the hindbrain is not a long cylindrical structure, and during development it's very clear that there is a huge flexion around about the midbrain level, and that tells a lot of the story. And Hiss drew these beautiful pictures showing the influence of this on the organisation of the brain. Number one, the midbrain ends up with almost no ventral surface and a lot of dorsal surface, but also the hypothalamus ends up rostral to the thalamus. It's situated underneath, but it's actually a rostral element within the forebrain. Okay, so that's the kind of background we've come to with this, and what we've attempted to do in the subsequent years is to take Lewis's chick brain ontology and merge it with our nomenclature set for the rat and then the mouse. And this involved some questions which we couldn't answer, and Lewis has put together the material appropriate for that. But we have got a rat and a mouse ontology, which was developed just at the time, developing just at the time when the BrainNavigator website was being developed by Elsevier. And Johannes quickly realised that the structure of the website had to be based on an ontology, and this seemed the right way to do that. And so he pressed us forwards to finish the rat and mouse ontologies, which are now present in the website. As I said, they're only published otherwise in the Chick Brain Atlas, and we're hoping to remedy that soon. We feel we need some kind of publication to let people know what we think is there now. And of course, we've also recently added to that, and George is going to speak a little bit about this in a little while, the rhesus monkey atlas with its nomenclature set and with the pictures which go with that. So I'll just take you through some pictures which give a little bit of a background. The reason we need a mammalian brain ontology is like a general conceptual one, and that is we want to relate the true relationships between parts of the embryonic and adult brains. So we have, in textual form, a fate map of the brain as it goes through. That's kind of a higher level scientific objective. But in doing this, we want to take advantage of the modern gene data of modern expression on modern data on gene expression, and the pressing issue is the informatics area, and that is that with large-scale informatics exercises developing, the need for an ontology becomes really acute. And you, in some ways, it doesn't matter what ontology you have, as long as you have one. That's a good start. But if the ontology could be one which was most logically based, that would be a real advantage. And that's why we've pressed on with this in an attempt to form a structure which would fit in with informatics projects. I only listed a couple of these because I've known Doug Bowden's NeuroNames project for a long while, and it's not only long-standing but also very comprehensive. There are others such as Loney, and as I said, underneath that we've slipped in the avian brain ontology of Lewis and attempt to take that forwards. But it's fair to say that all of the other brain ontologies are largely based on traditional classifications of the brain structures, which have been with us, the nomina anatomica kind of classification, and there's good reason for the things being in that way, and people who put those together had no reason to think that they would be challenged from an ontogenetic point of view. And you can still use any one of those ontologies for any neuroinformatics system if everyone is consistent through the data use that they make of that. This is really saying the same thing again, that the traditional systems used the conventional topographic point of view, whereas this is based on ontology. Because it's based on ontogeny, because of that, it actually applies to mammalian, avian, and reptilian brains. You can probably apply it to all vertebrate brains with modifications as you go back, because it does follow the developmental sequences that are common to all of those brains. And when you look at zebrafish brains, you can find rhombomeres and you can find the isthmus and you can find the same component parts are there. The extent to which you can identify all of the homologies is a bit of a challenge, and it gets tougher and tougher when you drop out of birds and go back towards reptiles and amphibians. But there's no reason in principle that it wouldn't do that, and that would be a big advantage if we could do that.